Okay, so this lesson we're going to be talking about cleavage, and it's going to be a pretty short lesson, but it just provides some nice background information and will help us further in our next coming lessons on like blastulation and gastrulation, because a lot of the next topics are going to connect and go off and further explain one another, so this will be short, but it provides some good background information. So just for some background and introduction information, so the process is going to go from blastula, which is just a ball of cells, to your gastrula, which is when the three germ layers are formed, the endoderm, ectoderm, and the mesoderm, and it'll eventually go on to form your embryo. So before all of that happens, you're going to go through cleavage processes with early mitotic divisions. So with mitotic, we know that's going to be diploid to diploid, in which you create those identical cells. So for that diploid cleavage, here is just a picture explaining the process. So we know your DNA is going to split to opposite sides, and then your centrioles are going to move to the opposite sides too. And then you have these microfilaments in the middle, and they're just going to shorten until they pinch off, and you have those two identical diploid cells. So this next slide is actually pretty interesting to think about. So here on the right, we have a picture of a homunculus. And so this just goes based off the perforation theory. So back in the day, they used to think there were tiny humans that were fully developed just in the head of sperm, and that's how development happens. But in reality, we know that's not true. In reality, we have the egg, and that develops, and so epigenesis is the actual process of how development works. Okay, and then just some important terms to define polarity and differentiation. So polarity is going to be differences in your regional and distribution of parts of the cell, and this is going to be due to differences in distribution of your mRNA, your proteins, and your pigment. While differentiation is when it develops your basic cellular function. So I think polarity has to do with more differences in structure and being able to identify those differences, and differentiation has to do more with function and having those basic cellular functions. So, for example, here we have these pictures over here showing the animal pole and the vegetal pole. So this is showing you differences in polarity because you can clearly see the different parts of the cell. Here you have your vegetal pole, which is thicker, and then this darker part is going to be your animal pole. So that's more dense with the proteins. So like we said, polarity is due to differences in distribution of proteins and pigment and stuff. So here you can see that example. The animal pole is where it's going to be darker and more dense with proteins. And I think that'll be all for today's lesson. Again, we'll have our quiz on our Instagram to test your knowledge and understanding of this topic. And then I hope you guys have a nice day.